chair this session. So most of the speakers, I think, you are all here. Okay. So my name is Yanis Bushtakuladi. I work with Peter Linder in Zurich. And this is a piece of study we started about 6 months ago on the fonioids. So it's these grasses, very charismatic to the Cape region in southern Africa and in Australia and South America. So we just firstly start with the picture of a very smart man who actually was the first to conceptualize what radiation means, and by basically implying that for having a radiation in a group of organisms, this particular group of organisms need to access a particular zone as an ecological opportunity, or what he would have called adaptive zone. Once there is this access to this zone, then you need to evolve particular traits, key innovations, firstly to be able to access physically this zone, but as well as to adapt yourself to eventually different ecological contexts. And now, with the use of phylogenetic trees, species level, if we're able to have 100% sampling for the group under study, then we have a very nice proof of macroevolution using phylogenies. And there are a whole set of different methodological approaches that people talked about in the first session. So with these diversification packages from Grabowski, the BAM, and these new statistical packages to be able to reconstruct traits under particular evolutionary models. So basically, using phylogenies and these new statistical approaches, we can identify models of evolution. And what I mean by models of evolution is that if we take phenotypic space and then time, we can say, well, okay, we have these two radiating biological entities, let's say, and then through time, they would evolve similar phenotypes, similar traits. You can have as well divergent evolution in which this particular biological unit would evolve completely divergent traits through time. And then you can have as well parallel evolution where these guys evolve in parallel and start with different ancestral traits and then evolve different derived traits. So basically, using phylogenies and all these packages, we're able to try to test which kind of patterns do we see. Quickly, we already talked about OU models, but it's just like a very simplistic graph. Basically, you have the traits evolving, a phenotypic optimum, and you have a rate of stochastic motion for this particular trait through time, and then a strength, a selection towards this particular phenotypic optimum. And this model is quite cool because it's possible then to define this phenotypic optimum based on a priori hypothesis. So basically, if we compare explicit models under an OU model with this package from Beaulieu, OUE, we can specify the number of different regimes and the location of these regimes on the phylogeny. Obviously, we need to have a priori hypothesis, a priori knowledge on what we want to test. So basically, if we take these three different models, we have a complete species level phylogeny. It's possible to select regimes on different parts of the phylogeny. So if I want to test for convergent evolution, let's suppose that these two clades here radiated. We have a shift in diversification. We want to test for convergent evolution, so we apply three regimes, one ancestor to one radiating clade, one ancestor to the other radiating clade, and then the two radiating clades evolve similar phenotypes. Same for divergent evolution, same for parallel evolution. So let's try doing that, because you'll see that it's quite problematic. We have a test case, our grasses. Again, it's very nice plants, mostly on top of mountains. That's picture from the Cape region. They dominate temperate open systems in Southern Hemisphere, South America, New Zealand, Australia, Southern Africa, with about 300 species, and we're able to sample now all the species. So we have a 300 species phylogeny. There is quite of a strong spatial variation in species richness among the continents. If you look at the map, we have 122 species in Southern Africa, which is the center of diversity. About 50 species in Australia, 60 in New Zealand, and about 50 in South America. A few outliers in the Northern Hemisphere, but let's not consider these guys. We run BAM, this is the phylogeny, and that's the start of our hypothesis, our explicit hypothesis. We found two shifts in diversification, one within the Pentamerus clade, which is typically Southern African, and one shift here within the Lithodosperma, which are Australian, New Zealand, South Americans. It's quite neat because if we look at the speciation rate through time, we see that at about, let's say, between 5 and 8 million years, we have an increase in speciation rates, and then a little slowdown. Then if we decompose the speciation rates by pruning the tree and only considering radiating clades versus the rest, we see that actually these two radiations explain most of the variation in speciation rates. So this is nice. We have two radiating clades. Parallel radiations, one in Africa for Pentamerus and one Australia, New Zealand, South America for the Lithodosperma at the Miocene-Pliocene transition. We know that the Miocene-Pliocene transition is characterized by an increase in seasonality, decrease of the forest cover, aridification, increase in fire regimes, especially in Australia, that has been shown. So now that's our hypothesis. We say these two radiations may be the consequence of convergent evolution. These two radiating clades evolve similar traits to cope with, as Simpson was talking about, an ecological opportunity or an adaptive zone. So okay, let's do that. Then we select traits that we think are potentially linked to these radiations. So we have the plant height, the number of spikelets. This is this very, grasses are very nice. The flowers are very, very small, but these actually are tons of flowers in here. Number of spikelets, this little reproductive structure in the inflorescence. Then we look at the actual flower, the own length, which is this little trait here that allows the seed to basically germinate on the ground. Then the number of florets, it's very, very, very tiny. The number of florets in every single spikelet. Then another trait, leaf anatomy, orthophily. This is like very, very kind of common structure we see, orthophilous leaves, so mostly like deciduous forests or things like that. And then sclerophyly, where you have leaves that are very tough and thick. So we basically looked at an anatomical section. We have about 100 characters. This is a leaf cross-section. And you can see that orthophilous leaves, this is all blue here, and this is all photosynthetic cells. Whereas here for sclerophyllous leaves, you see that there is quite a lot of sclerenchyma, so a lot less photosynthetic cells, and leaves that are very, very tough and hard. So this is what we did, and we used non-metric scaling and ordination axes to come up with a quantified character, a continuous character. And we did run OUE. So basically saying, well, for these two clades evolve similar traits. And we find that for height, for the leaf anatomy, and for several other reproductive characters, it seemed to have undergone convergent evolution. So these two clades seem to have evolved similar traits. For the number of florets per spikelet, however, it's more of a parallel evolution. So these two radiating lineages evolved different traits, divergent traits. Cool, so we have these two radiating clades. Remember the New Zealand, Australian, and the African one? And then if we estimate then the parameters, indeed we find that it's tending to a lower plant height, to a different optimum value, same for the anatomy. Now we have a problem, because we define these models with a priori hypotheses. Now we select models, we compare models, but the best model is the best model compared with the other model we selected. We cannot obviously test for all possible evolutionary models that, that these grasses have undergone. And then there's some issues as well with this very nice paper by Juan Ane about type 1, type 2 errors with this kind of thing. So, okay, let's now use this Uyeda and Arman package, which basically estimates the dynamics of the traits only from the data we have and the phylogeny. We do not infer anything. We do not have any hypothesis. We just want to look how the traits behave on the phylogeny, and only looking at the traits distribution and the phylogeny, and then that's become problematic because the best model is actually a model in which we not only have the radiating clade evolving a particular trait regime, but as well some of these early diverging lineages within this group. So it's quite tricky. Now, this may be wrong, or this may be true, but one way or the other, the only conclusion we can say is that there is a drying trend through the Miocene, that we have simultaneous radiations, but at least for one of the radiating clades, we may have a trend toward short life cycles, orthophily, smaller plants, and that may be a cause of aridification. You rather live shortly but making sure you reproduce rather than investing a lot, growing high and having very tough leaves. The evolution of convergent or divergent traits cannot be assessed simply by comparing models, because once again, you need to basically test for all possible models that evolution the signal that evolution could leave. So more traits are needed. We did not finish all the analysis with all the reproductive traits I showed you, but this is one of the way I would say that one could test for these different models of evolution, not only by knowing your biology, but as well as having very, very robust model testing, and not only comparing models in which you think that will happen. And that's all from me. Thank you.